And so what I'm taking away from this is there's a couple of different steps here, right? So the first is what I'll call just collection of the raw material. So you have to actually get the blood or whatever it is. Then there's an actual scientific process that is identifying the cells and kind of having that precision technology or science that you talked about. And then now what you're talking about is this third step, which is really just data analytics, right? It's very kind of high level. It's very large data sets, but it sounds like machine learning type problem where you ultimately can say, hey, we have confidence that we collected the samples from the people that we think we did. We've got confidence that we were able to use our precision technology to get accurate identification, kind of the raw data that we have we're feeding to the machine is accurate. And then if those machine learning algorithms and kind of computers are structured correctly, they should be smarter than the average doctor, right? And come back and say, here is exactly the path that this person is on based on the genomic sequencing. And then you guys almost are going to build like a recommendation engine after that then says you should go get this test or you should go do X, Y, or Z because of the machine learning has matched you to these other people. And that's where we know how to make that recommendation. Do I have that kind of right?
Yeah, I think you got very close. There are two more steps that make this whole thing work after that. The next step is clinical algorithms. And, that is something you cannot do through a machine learning system. You have to do this. You need a lot of people involved. You need to apply what's called a Delphi method. So you have to ask a lot of experts for their opinion. What happens under all these circumstances, right? For all these different cancers, what downstream clinical diagnostics should we apply? And here you cross the line from science into medicine, which is a very different thing. So medicine is all about, who agrees, who are other people saying the same thing, because you have to get to a consensus among experts, otherwise you're screwed, right? You need liability out of the way, you need to implement that. So it is a very different field than just science. The scientist can just say, well, I think this is the best solution because here's my statistics. In clinical reality, you have to say, but who are the people who agree with that? Because it's a much more holistic problem. And then the last step, so fourth is clinical algorithms that this whole thing needs to work with, which is very complex. That's one of the main reasons why smart tech guys who go into biotech often fail, because they lack the understanding of what goes beyond science and engineering, what goes into this weird clinical space, this authority and doctors and experts and legal and compliance. But you have to solve that, otherwise you don't have a product. And the final step is, of course, the business model. Like, who actually pays for that? And that, again, sounds maybe trivial to most tech and finance guys. Like, why is this? I mean, if you save people's lives, they just pay for it. Guess what? Not in medicine. So, we changed that fundamentally, but it gets insanely complicated. How do you convince a insurance bureaucrat that he likes that? And if you show that you save people, like, sorry to be so frank, but they don't care, right? That's not their job. You have to show them that they save money. And you have to show them that you save money immediately or quickly over time. And then, things get insanely complicated. And that kills also most of the innovation. So if you don't solve 4 and 5, all your amazing tech is not going to work because you can't finance it. And so this is like now I gave you the full picture and complexity, why it's absolutely non-trivial to truly understand your business model, to innovate on that front, and to be highly synced with the medical and clinical reality of things. And then nail the tech. And then you're getting somewhere.
Yeah, it's absolutely nuts. And I guess part of that clinical side, when I hear the word clinical, I think of all sorts of different tests that can be run. With this genomic sequencing stuff, is this only being done on humans? Was it previously done on animals? And then that's where the technology kind of was perfected and now it's being brought to the human side? Like what is the relationship there in some of that testing to non-human kind of clinical trials or whatever you could do there?
Well, the problem is, or the advantage of genomic sequencing is you don't have to do a lot of animal trials because there's no risk in just collecting the evidence, right? So that was very human driven from the outset on, Craig Venter and the Human Genome Project. So you can sequence, a lot, and you don't, without any kind of approval, because if you do clinical trials and you just sequence, no one gets hurt as long as there's no feedback loop. So it's very different. The actual sequencing and developing sequencing technologies, you're free to test this on humans because you're not putting anyone at risk, especially if there's no feedback of information. Once it gets into cancer screening, of course, the game completely changes, and that's the problem. That's what's holding the liquid biopsy industry back. So what we discuss is called liquid biopsy because it's liquid and kind of a biopsy. And that's where we innovated a lot to actually get out of that problem and get medical advancements and technologies to our members much faster than we normally would be able to do that if we just go through Medicare and commercial insurance.
Got it. And then I guess as you get better and better at this, right? Because part of the machine learning algorithms that are actually analyzing all of this sequencing, they should improve over time, right? It's kind of the beauty of machine learning. Help me understand, is that something where, let's say I'm testing for a variation that we believe could lead to a higher propensity for cancer, right? Can I use the advances and the accuracy that we've determined on that test in those machine learning algorithms to then apply to a different type of test? Like, is there some shared knowledge or shared analysis that can be moved from test to test? Or is each one of these tests, when you go to start them, they're really starting from scratch, right? So you have to rewrite the algorithms and kind of start all over again without any of that shared knowledge.
I mean, I'm super excited about these things because it's probably the most challenging and rewarding data science problems I've ever seen. Well, there are some in finance that are also pretty immediately rewarding, but I mean, in a human sense. And you can view it as like, in a way, if you take a step back, it's like any data problem, right? If you want to determine something, if you want to figure out if a certain real estate offering is like likely to increase in value over time, you could build a crazy machine learning thing around it, like how many people look at it, where it's listed, what the current price is, what the stock market does. Like, you could feed all that information. And how does the stock market data relate to that real estate as opposed to other real estate listings around it? Well, no one knows. These are two different tests, right? You combine these test sets, and if you have sufficient data sets, you can make them work together. And that's, if we test your hereditary, if you understand your hereditary variants, and we understand your somatic variants, the somatic variants are also like a time-based, kind of on a time series, because we do that every year. Whereas your hereditary, we don't have to do every year because it's not going to change. So you have very different types of data sets, and so you have to learn how to make them compatible. But each data set adds a significant amount of insight and dimension to the problem. I think there are two major challenges in medical data sets. The first one is they are often very heterogeneous. So if you have 1,000 or 10,000 patients, the probability that you have the same data sets for these 10,000 is like 0, unless you do a super clean clinical trial that costs you an immense amount of money. If this is commercial patients, then I might have your hereditary genetics, but not the next one, next person's. I might have your hereditary and your somatic and good clinical records, but for the next person, I might only have hereditary and some records. So when you build machine learning systems, it's very important to account for that, right? That you sample, the comprehensiveness of data is nearly impossible to maintain. And just cutting it down to the data dimensions that are comprehensive is kind of stupid because you're losing so much information. So you need much more fuzzy, systems that allow you to recognize much more things. And you also need to embrace more, man-machine hybrids, where you have experts and analysts sitting in front of these things and use the machine learning more as a decision support system and have these mutual interactions. That's very important in medicine. And then the ultimate problem, of course, from a business perspective is you need to close the business model loop in order to even get that information, right? You need to figure out, how do I actually make money and create a business model here? And that's very, very hard. And I'm very happy we solved it the way we solved, because for most companies, that means you have to just do very rigid clinical trials over a decade that costs you hundreds of millions of dollars and are super inflexible. And in the end, you have bad data sets because you missed out on the most important dimension. We see this with a bunch of our peers that just don't have the right data sets.